How loud do I have to talk? Can you guys hear me online?
Yes.
Okay. All right. So thank you all for listening to my talk today. I'll be presenting sort of mini research update where we've noticed some sort of atypical optical properties from Sp3 hybridized and Sp2 hybridized functionalized carbon nanotubes. So first, carbon nanotubes we think of like as if we have a graphene sheet, a highly conjugated Sp2 hybridized structure, which can be rolled up into a tube. And this is what we think of as like a carbon nanotube. However, these sort of pristine nanotubes typically have lower photoluminescent efficiency due to this high level of conjugation, which allows for really delocalized excitons, resulting in lower in energy optically inactive states for these pristine systems. So it turns out that introducing a defect onto the surface of a carbon nanotube, here I'm showing an Sp3 hybridized defect. Okay, an Sp3 hybridized defect results in actually lower in energy lying optically active states upon photoexcitation of these systems. So this is because the introduction of this Sp3 hybridized defect kind of breaks that conjugation in the highly conjugated system, which then can cause for the exciton to become sort of trapped or localized on this defect-induced state, and then have emission, optical properties of emission of their own. They're typically redshifted, lower in energy, and optically active in comparison to the states of the pristine tube. So something that's going to be kind of important later on in the presentation is how the tubes are referred to. So if you think about like a piece of paper that you roll up into a tube, you can roll it up into having all sorts of different diameters and different angles. And so you can do the same thing with a carbon nanotube. And so here I have, you imagine the big square being like your graphene sheet. You cut out a rectangle from that sheet and use that to roll up your carbon nanotube, right? So the sort of length of the vector defined as R on that image is like the circumference of the unrolled tube, and that can be broken down into these spatial vectors R1 and R2. And so the coefficients in front of these R1 and R2 are what are used to define or sort of name the different types of carbon nanotubes. So for example, in the image on the screen here, I have there's 6 units in the R1 direction and 3 in the R2 direction. So this would be a 6-3 carbon nanotube. The other figure on the bottom left-hand side of the screen shows sort of some of the many different types of carbon nanotube chiralities that can be conceptualized with the 6-3-1 circle with a little black circle. So I mentioned that the introduction of the Sp3 hybridized defect can result in some optically active states. But if you think of a double bond in a highly conjugated lattice, if you break one of those bonds to introduce a new bond, another position becomes available as well that also needs to be filled in order to prevent like unfilled octet, I guess you could say. So this introduces sort of 6 positions relative to that primary defect position that I call auxiliary positions that are now available for being filled. And so there's 3 positions ortho to that primary defect position and 3 positions that are para to that primary defect position. And they're defined, you know, they're differentiated with the plus, 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 minus symbols here, but it's not really important for you to be able to differentiate those as long as you know that those are different ortho and para positions that can be filled. So what I did here was I looked at kind of 2 different types of defects on the tube. I looked at this at the top. You guys can't see my mouse. Okay, I looked at the top left-hand side here with this N-methylpyrrole is what this type of functionality is called. And I looked at that in 2 different ways. The first way is where the one functionalization was bound to both the primary and auxiliary defect positions. And so this is what I call a bridged defect. And it's also I refer to it as an Sp3 hybridized defect because it is conjugated itself. It's extending the conjugation of the pi system of the carbon nanotube rather than breaking it. Compared to the defect, the functionalization right next to it with a hydrogen. So this just has that N-methyl pyrrole group found in one position and then a hydrogen in the auxiliary position. And then different kind of functionalization that I'm comparing to the pyrrole defect is just an amino benzene group with the hydrogen in the auxiliary position. So immediately there's kind of two major differences between the two types of functionalizations that are interesting and also pretty confusing. So it was confusing to me. So I should mention that the type of tubes I'm working with are 11-0 tubes. And if we go back here, the R2, the coefficient in front of R2 is 0. So there's no angle here. And so this introduces a sort of symmetry in the tube, which results in 2 of the 3 ortho or 2 of the 3 para positions become degenerate in energy. And so if you look here on this 11-0 tube, you can see that this blue square and the red square are aligned with the tube axis, which would be horizontal in terms of the screen. And then the triangle and the circle are both kind of parallel each other on either side of the tube. And so those two states I would expect to be degenerate in this case. Does that kind of make sense? Okay. So here, yes, then again. So let's start with the amino benzene functionality on the right-hand side of the screen. We do see the 2 degenerate states, which are the orange and the pink lines, the O++ and O- cases. And I'm only going to discuss the ortho configurations here because in terms of the bridged functionality, para is not really possible. It's too far away to have the same molecule bound in 2 places. But the pink and the orange lines are degenerate. You can see they're overlapping really well. And the non-degenerate case, the O+ case, this purple line, is very close in energy to the pristine case for this tube, which is the black line. And so here the non-degenerate case is blue shifted in comparison to the degenerate cases, and keep track that the O+ is the non-degenerate state. So now if we compare that to the pyrrole functionality, the non-degenerate case here, the O++ state is red shifted compared to the degenerate states. And the degenerate states are O+ and O-. So not only do we see a switch in whether or not the non-degenerate state is red shifted or blue shifted compared to the degenerate states, we also see a switch in which states are actually degenerate. And so that's kind of interesting, and it's tricky to explain. I'm not sure I can do it, so I'm definitely interested in what everyone has to say. But the first thought is to look at molecular orbitals. So these are just ground state molecular orbitals. And this, what I see here, I thought I had, yeah, okay. So what I see here is that for the conformations that are the most red shifted, they all have contribution in the orbitals from the defect itself, from the functionalization. So for the pyrrole groups, the first 2 on the left-hand side of the screen, there is electronic density on the actual functionalization, which is not the same for the degenerate states, the O+ and O- cases. And for the amino benzene cases, if you remember, the degenerate peaks were the most redshifted, and so these 2 are showing electronic density there as well. Another thing I looked at were the geometries, the ground state geometries of these systems, and a trend that I saw here was that the state that was not degenerate was showing a very different sort of orientation of the functionalization on the tube than the degenerate states. And so I thought that might be something else interesting to show you guys. Again, I'm really looking for any thoughts you might have in general about this strange feature we're seeing. Something else interesting is that This is actually a previously published paper. They did see this sort of trend previously where the energy of the non-degenerate state switched from being more redshifted or more blue shifted compared to the degenerate states. They did not see the switch in which conformations were degenerate, but they did see the switch in which one was more redshifted or more blue shifted. But this wasn't between different functionalizations on the same type of tube. This was between different types, different chiralities of carbon nanotubes. So the top panel here is for a 10-0 tube, and the bottom panel here is for an 11-0 tube. And so in this paper, they were able to see that they're able to sort of categorize carbon nanotubes into different groups based on what's called the modulus. And I'm not a big math person, but I think from what I read that this is like the remainder. Okay, I'm getting some kind of nods. So just think about it this way, it's just a different way of grouping carbon nanotubes. And so carbon nanotubes with this mod 1, where the modulus is equal to 1, are showing different optical properties, different characteristics than the groups where the modulus is equal to 2. And so these are referred to in the paper as mod 1 and mod 2. But for the mod 1 type systems, the non-degenerate case was more redshifted compared to the degenerate cases, which were closer in energy to the pristine state. And so I have the degenerate cases circled in red and the non-degenerate case pointed to with a blue arrow. And so for the mod 2 case, the 11-0 tube on the bottom, it's switched. The non-degenerate case is more blue shifted, closer in energy to that of the pristine state. So this is a very similar trend to what I saw, but instead of being between different tubes, it's between different functionalities. So I don't know, what do you guys think about that? They also showed ground state molecular orbitals in this paper, and the difference between mod 1 and mod 2 was whether or not the HOMO had electronic density aligned with the axis of the tube, which again is horizontal on the screen here. And so the HOMO had electronic density aligned with the axis of the tube for the mod 2 case, but not for the mod 1 case where it was switched. It was more like in the loop there. So
I have a question.
Okay.
So for your orbitals, which you were showing in a, I don't know, 5 slides, yes. We don't see this trend, right? We don't see a difference between amino benzene and methylpyrrole.
We don't see a difference in whether or not it's aligned with the axis of the tube in the HOMO. That's correct.
So I would say the explanation which they provide in this mode paper, I mean, when they compare different mod 1 with mod 2, looks like this explanation definitely not applicable in this case because we don't see change in the alignment of the orbitals. The orbitals are pretty identical in all cases, right?
Yes, I agree that it's not entirely applicable, but I do think the trend was similar, which made it stick out.
What is a trend? Trend in orbitals, or what kind of trend?
The trend in the non-degenerate state being more redshifted or more blue shifted.
Yes, the trend is identical, but I think explanation should be different.
Right, I completely agree.
Okay, good. Unfortunately, it should, would be nice if we had exactly the same explanation.
I agree. It would be nice to have any sort of explanation. But yeah, so in conclusion, we have these sort of unusual trends with the flip in energies between degenerate and non-degenerate states, between the two different types of functionalities. And I'm confused about that. Thank you.
Okay, let's continue discussing.
Yeah, Haley, can you again return back to your orbitals? And I just suggest to everyone find at least some differences between the three.
Similarities and
So, Helen, can you again repeat which are degenerate, which are not? Can you point?
Yeah, so for the parole case, the first two, the one on the left and the one in the middle, the degenerate cases are the ones that don't have the electronic density on the functionalizations. That's O+ and O-. And then for the amino benzene, it's O+, hold on a second. Confusing myself. It's shown better here. O++ and O- for the amino benzene and O+ and O- for the pearl group.
Okay, can you return back? O++. Yeah, so I think would be nice to this table to add the energy information, right?
Okay,
And this is not NTOs, this is ground state orbitals.
that's correct.
I also suggest probably take a look on the NTOs because you showing, of course they're related, right? But you showing excited state properties on your previous slide. You showing the excited state energies, and you talk about the alignment, and then you're showing the ground state orbitals.
Yes.
So once again, so for pyrrole, pyrrole, we have O+ and O- are degenerate states. And for the degenerate states, we don't have anything on the molecule. The density is sitting on the tube.
Right.
And in case of amino benzene, it is O++ and O- or O++ and O+.
It's O++ and O-.
And in this case, they have both on the molecule and on the nanotube.
Right.
You also were doing the analysis with these matrix densities. You remember, like this was on your poster.
Yep.
So is it possible to do same analysis for the case of methylpyrrole?
Absolutely.
Okay, just to compare how much, like, yeah, looking on orbitals, I also maybe not just ground state orbitals, but NTOs would be more reasonable to show. And I'm not sure that the paper which you refer, they show ground state orbitals. I think they show NTOs there.
I think it said frontier molecular orbitals.
Really? Okay.
That's why it showed the molecular orbitals.
Yeah, they probably will be not very different from what you're showing, but I think it would be, since we don't have explanation so far, right? And the explanation which is provided, the trends in the energy is similar, but we cannot really now use the same explanation as they provided for this mod 2, mod 1 cases, right? So that means that we need to really play with try to find another reason, right? So let's look on the transition orbitals matrix and compare all the three cases. Maybe we also will be able to better see the difference there. But so far the difference looks like what you kind of mentioned. The difference in the cases is involvement or not involvement of the molecule in the orbitals, right?
Right.
So for the most redshifted cases in the, which is degenerate most redshifted in the amino benzene case, they have contribution both on the molecule and the tube. And in case of methylpyrrole, it's backward. The state with a contribution on the molecule and the tube is least redshifted and it's not degenerate.
So,
and if you compare Sp3 versus Sp2 for this molecule, then they really very, very similar, right? Then there's really no any kind of difference so much.
yeah,
The trend is exactly the same.
right.
Again, like not orbitals which are not sitting on a molecule are degenerate and the most redshifted. Okay, good. Thank you. Yeah.
Okay.
Do you have any idea why they were classifying things by mod 3 specifically? Like why would it be the same? You know, if you have like 3 carbon rings, then you wrap it, and then 6 carbon rings, and 9 carbon rings. And why didn't they show the mod 0? I guess I'm just curious about like why it would sort of like why would you get the same characteristics every third ring of carbon?
It's a good question. I briefly read something about it where mod 0 was like metallic tubes. I don't quote me on that, but
I'm looking at the paper right now, and they say mod 0 is metallic, so they weren't considering it.
Okay.
mod 0 is metallic tubes, and then there's essentially 3 cases. I don't know if that's why they chose the number 3. I'm not entirely sure why they chose 3.
Well, I mean, if they fall nicely,
I missed.
there must be a reason for it. I just don't know what.
I'm sorry, I really missed the question. So why they choose what?
Modulo 3.
I think it's mod 1 and 2. There is no mod 3.
Right, but they're using mod 3 to figure out what their mod.
Ah, you mean the divider?
Basically, yeah. Like
Yeah,
why-
it depends on the geometry. I mean, it's so the geometry of the tube is completely dictated by the chiral vector and the ratio between n and m. Yeah, so you can predict what is metallic, what is not metallic, and so on. Like if you divide, yeah, if you have mod 0, I mean, if you divide it by 3 and it's completely dividable, then it is metallic tube. So it's the reason behind it is just like if you look on the geometry, right? So all these parameters just coming from the wrapping geometry of the tube, and because the benzene ring, the unit crystal, how it calls primitive crystal of the lattice is very symmetric and very simple, right? For the nanotube, it's just if you consider the benzene ring. So based on the geometry, you kind of can predict this metallic, non-metallic properties, and this mod 1, mod 2 is also just coming due to these angles, because again, you cannot really make any kind of angle, right? Because any kind of angle will not work, since you need to kind of connect them nicely, right? You cannot break the benzene ring in the middle. It has to be connected with each benzene ring. So that's why the geometry is very straightforward. And this rolling angle depends on the diameter and depends on your vectors A and B, right? So your chirality.
Yeah. They were doing strictly like, it was what, like 10, 0, 11, 0. It was always 0 for the second number.
They could do chiral tubes too,
Okay.
but since I worked on an chiral tube here, I just
Okay.
showed that.
I would actually put another question for your notation, since I thought, I mean, I understand we flip in the trend, but I thought from the geometrical point of view, means if your notations are consistent, I don't understand why your degeneracy is flipping.
I also don't understand.
So can you return to your structure? I mean, like how you show, not this one, but you show how you define your O plus or minus.
Oh,
Yeah, this one. So my understanding that degenerate cases should be triangle and circle, right?
that's what I understand to be.
So it's plus, plus, and minus. And this is the degenerate cases which we see with aryl, when you have aryl molecule, right?
Right.
Now you're saying that with spiral, are you sure you're not just changing the notations? Are you sure that it is really positioned in a square? What is it, square and circle, or which ones? Plus and minus now becomes degenerate for the spiral.
Right. Yeah, I swear I've looked at it time and time again, but I almost think I should just redo it to make sure.
Did the molecule in the O minus one in this case shift? Because it looks, just looking at the pictures, it looks like the O minus is O plus, like if I'm looking at, yeah, this slide.
This slide or which slide? Which slide to look?
Not right now is the one I'm looking at. So if you look at this one and this one, it looks like the ligand is facing the same way. Where it really should, the O minus should be more looking at it from the side.
Yeah, it almost seems like O plus plus and O minus are shifted,
Which
like switched.
my guess is probably you did notation incorrectly along the way, but
I hope that's the case.
that'd be my guess looking at it.
Yeah, it is very easy to check, yeah, because you clearly see where is your axis. You just need to check what would be these angles, what you call plus and minus, whether the angle with respect to the axis are the same as your plus and minus or plus plus in the case of amino benzene, right? Just recheck this. I mean, I think it doesn't change your overall results, but I think it's probably just misleading notations.
Okay.
Because from the geometrical point of view, it's completely clear why circles and triangles should be degenerate, right? because they're really symmetrically, like the structure is completely symmetric with respect to the axis of the tube. But if your molecule is placed on the position of the square, right, then, and again, with respect to the axis of the tube,
Yes.
they're not symmetric anymore with respect to the axis of the tube. They're symmetric with respect to the diagonal, if you take in like this diagonal rather than axis of the tube, but not with respect to the axis of the tube. And can you show X? So you're showing axis, right? But can you also show the chiral angle, like a vector, the chiral vector? Well, you using these figures from publication. This is not your own figures. So
Right.
can you really take your structure and really do this measurements and really figure out what would be the direction of the chiral angle? Axis of the tube, chiral angle, 11,0, right? So it should have directions with respect to the axis.
Okay.
And then exactly the position, maybe even measuring the angles and showing the angles with respect to the axis of the tube for each of your case. So in other words, don't do schematic, really do this in real. Take your tube with a given molecule, which already placed there, right, after optimization probably, orient it with respect to the axis, do the measurements, right, check what exactly the alignment of the defect would be.
Okay.
So I actually have a question about the last slide.
Okay. Right.
Not the little last slide, but
This is the last slide.
the last one that we were on with the showcase.
This one?
Back further. Well, that one, this one.
Okay.
Is there a reason why the amino benzene doesn't also form a two-bonded structure like in methylpyrrole? And is there also a reason why the hydrogen is closer to the methylpyrrole than the amino benzene? Or is that just?
That's just my bad.
Okay.
Yeah, then I just made that chem drop. So don't worry, the distance is,
Okay, just abstraction.
yeah.
But yeah, the first part, is there a reason why the amino benzene doesn't?
I want to say that I have seen RO groups in this sort of bridged functionalization, but that's not what I studied.
Okay.
Well, the idea behind amino benzene that it's attaching there through the diazonium salt, right? Which means initial geometry is, I mean, your initial molecule, the reaction goes not really with the benzene ring, the reaction goes with whatever the exact name of this diazonium salt. So it has a triple bonded nitrogen attached to the place where the benzene now is attaching to the tube. So, and this nitrogen is gone, creating a radical, and then radical attaching to the surface of the tube.
Okay.
So the reason why amino benzene people believe it's attaching through Sp3 and there is not possible to get Sp2, it's just the way how the reaction goes. People know this reaction.
One can imagine many attachments, but not all of them were realized experimentally. Challenging.
Yeah. So in case of amino benzene, the feasible alignment would be through the Sp3 bonding, right? Not Sp2 like with pyrrole case. But for pyrrole case, they believe that reaction allows attack. Well, actually, they're not 100% sure, but chemical intuition is suggesting that actually it's probably likely to be Sp2 rather than Sp3. And we tried Sp3 just for comparison.
So more questions in the room? More questions in the Zoom? Well, we will soon get to typed questions for a couple of them, but I want to ask London to serve as my translator. Okay, so I'll ask a question to you and then you ask her, because I fairly do not know how to formulate it. Maybe you will assist. So if you look on this slide that everyone likes so much, it looks like someone took it's either like corn, or someone took a tube and started to put toothpaste or Coke there, right?
Yeah.
And it looks like this two anchoring groups can be placed into either same thread of the pulp or in the different or at some angle.
Okay.
And, maybe placing them in the same or different would be the determining factor between the symmetry of orbitals and the level of redshift.
Okay.
So now it's try to ask Haley if she sees any connection or any correlation between attachment of the ligand and orientation of orbitals.
So I guess, did you get the question? For starters?
I think I get what you're saying about the corn cob. I think that I would probably need to sit down and stare at it for a solid 10 minutes to be able to make a good statement about it. Did you see something?
I told us, I've told my first.
Okay.
Well, the question is if they are going parallel or perpendicular to this space trace, which one gives stronger redshift? Parallel or perpendicular? This is
No, but Dmitry, parallel, it's HOMA, right? It's occupied orbitals, they aligned parallel. And perpendicular, this is alignment for the LUMA.
yes,
And
and 90 degrees difference between HOMO and LUMA, but still, can one? It is a hypothesis. Is there any correlation between this orientations of the orbitals and placement of the
Yes, if you look on it very, very carefully, right? So look on O++ for the case of methylpyrrole. So and look only on LUMA. So the bulbs on LUMA are like little sausages, right?
Duo Sp3, which there are two methylpyroles.
Doesn't matter which one. You can use, they are pretty much alike on both cases. Just either there or there. Let's look on the sp2 methylpyrrole on a O++, which is not degenerate in this case. Yeah, and you see that, how would say, they are not cutted by half, right? And if you compare with O+ and O- for the same case, then you can see that the orbitals kind of cut it by half. Yeah. Again, it's hard to explain what exactly it is, but if you look, you see the difference. And if you look on the Amino Benzene, it's completely reverse pattern. So those which are degenerate now have not divided, not half orbitals, but like tried orbitals.
Nodal plane between attachment points. Right?
Yes, nodal. Yeah, I like the way. It's a nodal between different colors, what we call plus minus, or it's probably not plus minus. It's imaginary real paths, right? Because it's a wave function. Not so the nodes between these different colors. The arrangement of these nodes is flipping when we go from Amino Benzene to the pyrrole.
Okay, yeah. It partially addresses this criticism. Haley, would you try to follow this?
And probably again, this difference in the nodes alignment also probably is reflected on the previously stated observation that if it is like half-half, then the orbital is not spreaded to the molecule, right? So change in the nodal pattern is affecting also how much it is hybridized to the molecule, how much it is spread to the molecule. And then we clearly see the trend. So we don't see the trend in a change parallel to the perpendicular, means homoluma kind of trend, right? It stays the same. It's reverse comparing to what we saw with the mod 1 and mod 2, where they just what was parallel now is this, right? So we don't see this trend, but there is still some trend in the luma. So I think we probably can address this difference through the similar ideas that somehow the alignment of nodes are flipping due to this different molecule attachment or type of molecule is changing the pattern of the nodes on the luma in these cases, which results on a flip between degenerate and not degenerate cases.
Thank you.
Did I address your question? This is what you're trying
Yes, you did.
to indicate us?
Essential part. And we do have an idea for Science Olympiad or outreach program for kids. Model orbital space. Okay, everyone is welcome to ask more questions. If no one is raising hand or raising voice, let's read the.
You show the absorption spectra shifting.
That was the question.
Yes, I just wanted to, because I was looking at the orbitals and
Okay,
I wanted to be able to compare the spectra shift.
I like those kinds of questions.
But Haley, so I think probably as soon as you're done with your second year exam, we will set up the meeting with Los Alamos group and maybe even invite experimental people, right? Similar to what you also participated in this meeting, right? Last time with experimental people. So you really need to polish your presentations. You need to, like, for example, change the line dashed lines for SP2 and solid lines for SP3, for example, right? Also put probably them to be easy to compare which one is which one. And in your orbitals, add the information about energies. So we really know, oh, this red shifted, this is blue shifted.
I absolutely want to make my figures prettier. And it's not that I don't like you guys, I just didn't have a lot of time.
No, I understand. That's exactly why I'm telling you that. Don't forget that you will have pretty much the same story, right? In a couple weeks for experimental people to prepare. So don't forget to polish your slides. Don't think, oh, I have it already.
On that note, it is very bright in this room, so that is affecting it, but I cannot tell the difference between the sticks for any of the dark colors. It just all look black.
So maybe we'll do dashes.
Well, maybe there's a shape on the top that's
Okay.
bigger. Actually, yeah, use the squares, the triangles, yeah.
Right.
Yeah, and again, if you use, for example, circles, squares, like in your previous slide, you were showing different symbols for your O plus or minus or plus plus. Yeah, here. It makes sense then to use the same symbols in the other, in your other figures where you use this same notations, right, to differentiate between this O plus or minus and so on.
Sounds,
Yeah. Makes sense, right?
yes.
But this is all kind of, format polishing. You just need a little bit more time to work on the slides, and then they will be perfect.
Okay, let's focus on the questions in the chat line and see any of them make sense.
Okay. So David asked, why is the meta position not a possibility? So my answer to this is that even with functionalization of RL groups, the meta position is not preferred. And we learned that in organic chemistry. But even with ortho and para positions, the ortho position is the most, I think, common because you don't have to break two double bonds to get the primary and auxiliary positions. So experimentally, it's ortho that's the most seen.
See, your stuff. That's pretty nice. Okay, I'm sorry to ask that question. You were basically, you had to pump it with argon, and I'm frankly never got an answer.
So typically we also on Chiron to switch your reaction aspects.
Okay.
Quite
The
small
singular
configuration.
triplet state. He's a good person to talk to.
I've asked him as well.
In the meta position, I think you break in the sp2 hybridization, not only in a position where this bonding is happening, but you also cannot preserve sp2 hybridization on the tube or for other neighboring rings, right? Well, again, just from the geometrical point of view, think about how you can preserve double, single bond.
Can one say that both ortho and para do have odd number of carbon-carbon bonds between initial and final, and meta would have even two of them. And then this symmetry difference is really became. Let's keep.
I'm sorry. Dr. Klee says sp2 and sp3 types of defect in the methyl parole. Please summarize the qualitative differences in their spectral signatures, which of them has a smaller total energy. Let's see here. The sp2 defect is showing larger energies.
So they're less favorable.
I believe so.
Okay.
Okay. Okay.
No, I would make a different statement here. So technically from spectral features, because we cannot really, the difference between sp2 and sp3 peaks in this case, for the most redshifted, is more noticeable, but still small, right? Because check, like about 10 nanometers difference here. If you convert it to electron volts, it would be really probably a very, very small difference. And in case of the more blue shifted transitions and which are degenerate for both cases, the difference is even smaller, right? Almost impossible to differentiate. So I would say from spectroscopic features, there is no way to differentiate between sp2 and sp3, unfortunately. Would be really great if we saw a big difference.
You cannot say whether they have sp2 or sp3.
Just by spectra, I don't think we can make this conclusion. Well, if we believe that our data are correct, then according to our data, this difference is probably not possible to really use it as a, the difference is too small. I don't think that it can be used as a good resolution for these cases.
Mm-hmm.
And also, it's also possible to imagine that maybe, total energy, by the way, Haley, have you taken a look on the total energy of these cases?
I have not.
Yeah, because this is, you optimize the geometries, you immediately have these energies. So this is the first thing to compare, which of them thermodynamically is more favorable, right?
Okay.
Either it's sp2 or sp3, and how much the difference is significant. Because if the difference between your ground state or ground state energies, total energies, is also in the order of kT, then it means that both species also possible to be presented, right? And then this redshifted peak would be probably just twicely more intensive, because either this or this contribution will give you pretty much similar energy with a little bit increasing just a little bit line widths for the cases.
Okay.
And another thing which I want to point, experiment, they have not working, the experiment was done on 6.5, which is mod 1 tube, and 10.0, which is also mod 1 tube. And they see, in case of 10.0, they see very significantly enhanced redshifted emission for the 10.0. And they really see just one nice emission peak, means almost no any other samples, right? So I think in this case, if we get into the mod 1 instead of mod 2, we also probably will, with spiral, right? We probably also expect that it should be shifted. Same as it was shown for 11.0, 10.0, with a real group, they were shifting. Right? What was most redshifted is less shifted for one mode, but more redshifted for another mode. I think if we do calculation at 10.0, this, it also will be swapping, right? But it will be swapping now in a sense that degenerate probably will be the most redshifted. And then again, if we have total energy not very different between sp2 and sp3, and we can conclude that both sp2 and sp3 probably can take place, but their energy is pretty much similar, right? Then they will see this peak coming from 4 species simultaneously. Means it would be really peak and a very similar energy with a very high intensity, because 4 species give exactly the same energy, give exactly the same emission from the same energy. So it's really important to calculate 10-0, and I think Brandon answered to us, right? Haley, can you give a short update about 10-0 calculations? Did you start working on 10-0?
I've already begun the optimizations. Now we're just waiting.
Right. Okay. But you already have 10-0 with aryls, with benzoaryl or whatever. Yeah.
Yeah, with some kind of weird oxygen on it.
Oxygen.
I'll show you later.
Oxygen instead of hydrogen.
Yeah.
Ah, okay. Yeah. But again, this change is not very, I don't think it's changed very much, but you probably also can change it to hydrogen to be consistent, right? And re-optimize the structures from Brandon with hydrogen instead of oxygen.
Okay.
That's what we already discussed. What about toothpaste?
Okay. And then one last question about degenerate versus non-degenerate. So degenerate means that there's 2 configurations that are showing really similar or the same energies. So on the absorbance spectra here, like the one on the right-hand side, the pink and the orange lines are overlapping almost perfectly. So I would call those 2 states to be degenerate.
Yes.
Compared to the purple line, which is not overlapping. So that would be the non-degenerate case.
Yes, but it also goes with the symmetry, because your O++ and O- are completely symmetric with respect to the axis of the tube from geometrical point of view.
That's right.
But O+ is not, right? O+ is different because O+ is sitting exactly on the axis.
Yep. So if this is the axis of the tube, the triangle and the circle are the degenerate cases, and the square would be the non-degenerate case.
Mm-hmm. So that's yeah. So that's exactly the question is, if your notations are exactly the same, then it's really strange that we have in not geometrically symmetric cases to be degenerate. So probably, yeah, there's just confusion coming from the notations, which you have to recheck.
Great.
Okay, let's make a countdown. More questions to Haley once.
I have one last question, and I'll make it quick. So like with all the suggestions that Dr. Kalina has suggested, like what is like the practical steps? Because I guess I'm missing out on what I'm hearing in general is that it's possible her, let's say optimizations haven't been correct, or am I misunderstanding that's what we've suggested? What is the practical steps next? Yeah.
So in terms of one of the cases where the degenerate cases had not the same conformations, and so the idea is that maybe I just didn't label them correctly. Like the calculation.
It's just naming problem. It's not the problem with the geometries or with the results. The results are trustable, but naming is probably was done incorrectly, not consistently, because names, we just made these names. Yeah, you can call it forever. You can call it Svetlana and Dmitry instead of O++ and O-. So naming means nothing, right? You just need to be consistent with naming, and it looks like this not happening with Haley case. She probably switched the names in one case comparing to another.
Okay, thank you.
So this is what she has to recheck to be sure. Well, if we are right, if it's really just naming problem, right, then it's a very easy fix. If it is really that states which are having not geometrical symmetry, but somehow we see that they becoming degenerate, having very similar energy, if this is really the result, then we probably need to think, because then it's becoming suspicious. Since there is no any reason for why the degeneracy is not broken if we change in the geometry, right? If the geometry is not symmetric. Yeah, but more likely just naming question rather than actual physical results that geometrically asymmetric cases showing the same energy. I don't believe that this is really the case.
Let's continue
Thank you.
our option and make countdown once again. More questions to Haley once. More questions to Haley 2. More questions to Haley 3. Let's thank Haley once again. So next presenter is Hanmant. So Hanmant, please start sharing your screen with your post. And everyone is welcome to provide constructive feedback, type in questions. So the talk will need chemical intuition of all of us for interpretation and suggestions. Okay, Hanmant, please go ahead. The floor is yours.
Are you able to hear me, Dr. Gilly?
Swap displays.
Yes. Can you hear me now?
Yes, we can hear you.
Yeah. Okay. Hello, everyone. Today's topic is.
Can you please swap displays?
Right now, we have the presenter's notes, not the actual presentation.
Okay. 1 second.
Yeah, swap presenter view here. First option, yes.
Okay. Okay. Sorry about this. Today's presentation topic is ab initio study on magnesium interaction with integrin alpha wave beta 3 at ligand binding site. So basically, this topic I already selected for my course project. So I'm continuing that topic for further study. So initially, I will start with the introduction because here we are. Here we considered integrin with the metal ion as a magnesium. So basically, what are these integrins? So these integrins are the transmembrane protein. So these are in the red colors, red and blue colors, and basically it is on the membrane of the cell. And these integrins plays very important role during the cell adhesion, proliferation, and differentiation. And basically, it is attached to the extracellular side. Sometimes some of the integrins are attached to the another cell integrin. So these integrins are basically made up of 2 dimers, blue, which are shown in the blue and red color. So blue color is alpha dimers and red one is the beta dimers. So there are total 18 at alpha and 8 beta dimers, and they form total 24 different integrin. And in initial stage, these integrins are in the deactivated state. So it is in the bending form. So once some ligands from the extracellular side come and attach to the integrin, then it get activated from the bending to the unbending transformation takes place. And sometimes from the inside intercellular side of the cell, the taline will attach to the tail end of the integrin. So once again, at that time also these particular integrins get activated. So normally what happens, these integrins play a very important role during the cancer metastasis. So when at a tumor site, when the invasion happens, these tumor cells are separated, and when they travel to the extracellular site, it gets attached with the integrin. And this because of this attachment, these particular cells are then entered into the blood vessel, and then it travels to the another site. Once again, it is attached to the integrin, and then it is squeezed out and they form the tumor at another site. So when you see this particular transformation, the attachment of the integrin plays very important role. So this attachment is based on the activation of the integrin. So when we consider this particular integrin, at the ligand binding site, there are metal ions that promote the activation of the integrin. So here for our studies, how these particular metal ions are playing important role in the interaction with this ligand binding site. So for that, we selected some amino acid, we selected alpha V beta 3 integrin, and based on that, we selected a ligand binding site for the analysis. So first, I prepared the model for it from this particular ligand binding site. From the metal ion, I selected this particular amino acid around 10 angstrom radius. I prepared the model for for convenience. I am saying this amino acids as in a protein with the in the presence of MN9 as in a protein MN9 model. And after preparation of this model, I just deleted this particular metal ion from it and I created the protein model separately. So for these two models, I started the DFT calculation. So basically, when I check this particular metal ion model, protein metal ion model, that time what I observed that this particular metal ion is having a octahedral geometry. So based on that, the amino acids which are nearer to it, so I just prepared the names for it, and for particular items, I just give the name and I find out the distances and angles for it. And based on that, we come to know that it shows some octahedral geometry around the metal ion or manganese ion. Then I started the study, and initially we started the study with the PBE functional, and I run this calculation for the protein model as well as protein ion model, and also for the ground state, as well as I consider the excited state for the protein metal ion, where here in the density of states plot, what I observed for the protein model, I observed the symmetry. But when we introduce the metal ion into it, the symmetry goes away. So these things happened because of unpaired electrons in the metal ion, in the manganese ion. And when I checked the what you can say HOMO and LUMO orbitals for it, so here for protein model it shows the same HOMO and LUMO orbitals for alpha and beta spin. But when it comes to the protein metal ion, it is different for the both the for alpha and beta spin. So as well as here, if you check the HOMO-LUMO gap, it is also changed for this both the mod for the both this model. So for the metal ion model, manganese ion model, then I plotted the different charge orbitals, and I compared the charge orbitals with the shapes. And here what I find that the where I observed that this particular energy levels of these orbitals, it is showing the octahedral crystal splitting. And based on that, Dr. Klein suggested me to go for the excitation for it. And when I applied the excitation, where n up down, I applied the condition n up down is equal to 3. That time what I observed here, and once again I plotted the orbitals and I compared the energy levels for it. I compared the shapes for it, and here I observed the contrast in the energy levels as well as in the energy levels as compared with the octahedral splitting. And based on it, we come to know that there is a tetrahedral crystal field splitting which we observed here. So now for these three models, then I plotted the absorption spectra, and then based on the results of the metal ion and excitation metal ion model metal ion model and the excitation model, based on that, the result predicted that there is a still the results what we are getting here, it is what you can say, it is not a perfect, and the excitation energy what it was showing, it is underestimated. So for that we continued our what we can say calculation for different models for different functionals, where I considered the GGA U functional and hybrid functional, HSC functional, HSC 06 functional. So here I plotted the orbitals density of states for it, and as well as for the HSC function. So here this is the comparison for these three functional, where what I see that for the PBA and GGA U functional, there is a the gap what we are seeing, it is not that much different. It is not that much, but when it comes to the HSC or HSC hybrid function, that time the gap is much more. And so it is same for the protein model as well as protein metal ion model. See this particular gap which is tabulated here. So if you check the values for the PBE functional and HSE functional, so there is a much more difference what we observed for the HOMO-LUMO gap. But here, when it comes to what we can say GGA-U functional, it is not that much. I just need your thought when I will show you the absorption spectra, which mechanism we need to consider for this particular model. So here then I plotted the absorption spectra for it, and here it is the comparison for all the absorption spectra for all the functional. So here then you will see the absorption spectra for it. So here I observed that still the values which we are getting from the GGA-U functional, it is still underestimated as compared with the HSE functional. I still need to evaluate I didn't find out the absorption spectra for the excitation state. Once I will complete that particular part, then I will make some conclusion about it. But still, it will be better if you provide me some guidance on it. So how do I need to go through it and how do I need to compare? And then for the transition energy, I started the MD simulation, and initially I heated the particular protein model, and then I did the MD simulation for 1000 femtoseconds. So this is for the protein model. And this one is for the metal ion model. So then I plotted the Kanshav orbitals for the MD trajectory. So here, if you see, there is some energy gap what we observed, but here I didn't find, we didn't get the energy gap for the protein metal ion model. So as Dr. Kilin said that for the dynamic condition, can we get the similar kind of splitting? So I just considered one item from the ASP aspartic acid amino acid, and for that I just checked the distance. But as MD simulation goes on, the distance goes on increasing. So I don't think so we will get the same crystal splitting after the MD simulation or at the dynamic condition. So thank you. So this is all which I have for the presentation.
Okay, please join me in thanking Hanmant. It's very brave work in area where initial calculations are rarely applied. So let's go to the discussion section. Other questions in the room?
So it was covered by the Zoom things, but go back to the absorption spectra. One more. Yeah. What is your third function? HSE. Okay, thank you.
Yes.
How does the GGA-U functional compare to the PBE functional?
What Miss, what is the Miss?
Was your question answered? Yes. Okay. Yeah, Hanmant, go ahead answering to Hanessa.
So can you repeat once again? I didn't get your question.
How does the GGA functional compare to the PBE functional? Like why do you use that for your calculations?
It's a provocative question formulated in a little non-standard way. PBE is one of the GGA functions.
Yes.
One can contrast. Hanmant is using GGA plus U, which is additional correction. So maybe his GGA was also PBE plus U, right?
Yes.
Make sense?
Yeah, potential energy. Is that you?
Well, it's the next question. Like, but it is correction for spin-spin interactions, which is added artificially, not ab initio. correction. Okay. Kadas is sufficient? Yeah. Questions in the audience?
So the molecule that you were modeling, that's partially ligand and partially protein that you were looking at?
Yes.
Where was the split, I guess? Like what was the, like what was protein and what was ligand there?
Means, are you asking where is the protein?
Yeah, so like in here, basically like which part of it?
Yes, yes. So for convenience purpose, I am saying the protein because the integrin is a protein.
Yeah,
So when I selected the particular model, it means when I created the model, I selected this particular site,
yeah,
and from this site, what I did means around this particular manganese ion, I created a what you can say. I selected the particular residues around the 10 angstrom radius, and so this particular amino acids means the structure whichever means those amino acids which are under what you can say within the 10 angstrom radius, that amino acids I selected for this particular calculation. Other than that, I can't. For the
and then you're using the same one in your picture on the left too?
uh,
Makes me see a model or a molecule that looks like the same thing, but I can't really tell if it's actually bonded or not.
I didn't get.
It's the same ligand for both systems?
Yes. Yes, I just deleted the manganese ion from it.
Okay. Okay, let's check if there are questions from online attendees. If anyone wants to challenge presenter or provide some constructive position. Okay, if no, let's Hanmant, will you please look through typed questions?
Okay. Yes.
I'm sorry for asking too much. I was just excited by your presentation. Please go ahead. So,
Okay. Examples of extracellular matrix are you asking? Dr. Kelly? Hello?
yeah, just give an example.
Means extracellular matrix means you can say that in our means under the skin, if you go means if you in our body under the skin, what you can say, you will see the bloods, and in the blood you can see different what you can say muscles and other things. So
So it's organic tissues. You are not depositing proteins to inorganic materials. It's different biological tissues, correct?
yes, yes.
Okay, yeah, thank you.
How does MLI? So is so Dr. Kilin, is there anything you need about it, or can I go to another question?
Yeah, let's go to another question.
How does MNI contributes the structure of its? Yes, I think when you check the can I read the question, or
You can, yeah, please.
yeah, how does metal ion contributes the structure of its vicinity? Does it break any bonds? I think in MD simulation, if you check that, sorry. So here, if you see the manganese ion, it is already broke some of the what you can say bonds for the residues. So I think so they break the bonds.
Okay, is it good or bad? Is it something that one would expect or?
No. I think, yes, I am expecting the same thing, but I didn't expect that it will break the bonds, but I need to check. Because I am doing the MD simulation also. So that time I didn't observe any breakage of bond or any formation of the bond with the manganese ion. Maybe I need to check once again that I need to check the after hitting what actually happened to the what you can say to the geometry of the molecule. So once I will check that, maybe I will get idea about it.
Okay, thank you.
Highly likely there will be questions, suggestions about density of states for this model from the audience. Okay.
So there were no, but I think when you present your talk once again to this or different audience, someone will ask, did you compute PDOS? You'll just ask to show partial projection density of states.
Okay.
Just consider doing it and consult with other group members who are doing it on a regular basis.
Okay. Okay, I will include.
Make feedback.
Okay, let's keep going.
Is this right that upon changing the magnetization from 5 to 3 coordination pattern changes the octa to tetra? Yes, I observed it for when I use the PB functional, that time I observed. So where I compared the orbital shape and the energy levels, and where I observed the contrast for the energy level. So I think for the PB I observed, and also I observed the same, I'm not saying tetrahedral splitting, but I observed the octahedral splitting for the GGAU functional. Maybe I need to check it for the HSC06 functional.
Okay, thank you.
Hi.
Yeah, you showed the crystal field splitting in the schematic diagram, but you didn't calculate it.
What?
You showed the crystal field splitting in your schematic diagram,
Yes.
but you didn't calculate, right?
So I just checked the values, energy level values. And I just compare the shape. I just check the shape around the manganese ion. And based on that, I just compare the shape of the d orbitals. And based on that, whatever the orbitals whatever the orbitals I considered for that orbitals, what is the energy level I checked, and based on that I make the
The energy levels and just leave it, and what is the energy level? Didn't you put it down?
here.
Didn't you wrote the energy level values for different d orbitals, like dx square minus y, dxy, dxc? This will give you the crystal field splitting between two.
Yes. What I did, let's say I started with the homo alpha minus 1. For this orbital, I checked the energy level, means what is the value of the energy. I just checked it, and so I checked the shape of.
What energy value you checked? You checked
So,
the homo alpha minus 1, homo alpha.
Yes.
Yeah,
So do I need to check any other value?
yeah, you mentioned in the left dx square minus y square is the schematic presentation.
Yes.
You didn't wrote the value of energy for this level.
Energy values for?
Not for this dx square minus y square, dc square.
Are you asking on the diagram on the left or computations on the right?
Okay, Miss, I didn't.
Let's check with Dinesh what he's asking.
Diagram on the left.
So diagram on the left, it looks like it was borrowed from a text.
Yes. Sorry, I didn't put the. Sorry, I didn't. Yeah, I didn't put the reference for it. So I borrowed it from the book.
So one is not expecting to have one-to-one reproduction of this diagram in the calculations, and only the way to guide the eye and
I'm
see
sure
what
if you count those events again, so.
it will be a lot of changes compared to an idealized.
Also,
idealized case.
Another question from me, how did you determine the U value for manganese?
What?
How did you determine U values for manganese?
Oxidation number are you suggesting?
No, this is you employ GGA plus U, right?
So here,
Yeah, right.
magnet,
GGA plus U,
huh?
you choose U value for manganese one, right?
So Hanmant, when you run calculations for DFT plus for DGA plus U,
Yes.
input files like incar must include numerical value for the U parameter. And Dinesh is curious which numerical value for the U parameter you use, something like 3 electron volts or maybe something different.
I forgot, but I need to check. Oh, the program I just whatever the program which is already available there, I just use that value directly there. I didn't check which value is there. So I will check and I will let you know.
Okay, thank you.
Okay, let's keep going.
GGA.
No, you already answered it.
Yes.
Next question.
How did you set your values?
Yeah, you already answered it.
All tables reports E. What about?
It is a suggestion. You don't need to
Okay.
answer it now, but reporting cross spin flip excitation gap will be relevant.
Okay.
Because when one excites system is manganese 2+, the actual excitation often is associated with spin flip, which means one promotes electron from alpha to beta, and this beta to alpha gap could be of interest to the audience.
Okay. About movies, where the amount of vacuum, it looks like model is expanded. That is the thing which Dr. Killian about the as you observe that the molecules are the atoms are moving out. So I don't. How do I restrict? How do I already whatever maybe I think I. When initially I created the model, that time I used the 666 angstrom, I think, value.
So you may try to use much less to have no vacuum or vacuum comparable to bond distance, like maybe 2 to 2, and see if it will give qualitative difference. If we would have unlimited numerical power, one probably could put it everything in a water box to prevent this expansion.
Okay, so can we do it? So for
Measurable.
Can you repeat once again, Dr. Killing?
The expansion of a model that you observe in the movies could be an artifact that doesn't happen in a realistic experimental situation. So one would need either to have less vacuum to have a constrained size of the box, or put a lot of water molecules around.
Okay, so for that, okay, so so can we add that thing during the MD simulation, or we need to do it from the initial stage?
For consistency, one would need to do it for everything.
Okay. Okay. Okay, I will do that.
Thank you. Let's keep going. Last question. Oh, there are more questions. I see one in the audience, maybe there are more in the Zoom.
Do you compare different multiplicities, or do you use n up down free for all? If so, how does total energy compare? Basically, I started with the 1, 3, and 5, and I observed the tetrahedral splitting for the n up down 3. That's why I just concentrated on the n up down is equal to 3.
Can you restate that again, quick? Why did you concentrate on the n up down equals 3?
So basically, I ran the simulation for n up down is equal to 1, 3, and 5.
Okay,
And then I just compared the energy levels for all the. I just plotted the results, and I plotted the energy orbitals for it, and the crystal fitting splitting which I observed, which I observed it for the octahedral splitting. I observed it for the initial model, and tetrahedral splitting I observed it for the n up down is equal to 3. So that's why I considered the same model for the future one. So do I need to calculate it for the 1 and 5 separately to compare the energy? I don't know.
so you determined to use n up down equals 3 based on the crystal field splitting is what you're saying?
Yes.
Let me intervene and share slightly different opinion. Same thing, just different logical direction. Hypothetically, upon photoexcitation, the spin flips and it changes from n equals 5 to n equals 3.
Okay, that makes sense.
And then Hanmant did perform optimization for this excited state with n equals 3 and found that this change of the electronic state induces reorganization of oxygens near the manganese ion.
Okay, that makes sense to me.
Okay,
I was trying to figure out why you were looking at n up down equals 3 because that's a spin flip excited state generally.
Yes.
As a simple question related to the crystal field splitting. Are you sure that it's not dehedral? Because the structure that you showed next to the diagram looked more like a trigonal bipyramidal, which is a possible organization for manganese plus 3.
That brings into another point. What are we sure that the oxidation state of manganese is +2?
Yes. Because the structure what I considered it is based on the experimentation, and it is already available on it, and I use the same structure for the for my study.
So is your model charged? Is the ligands or protein or whatever we're calling it, is that -2?
Yes.
Okay,
So ligand, the ligand, the protein model which I have, it is not any charge, but the ligand which it is attached to the attached at the integrin site, that one has some charge. Maybe because of it, that manganese ion, it was showing the +2 oxidation state.
but
so it has an overall neutral charge.
Yes.
Does the model that you were looking at when you remove the manganese then have a -2 charge?
So I didn't check for that, because in the MD simulation I use the whole geometry for the simulation.
And you should do PDOS.
so Hanmant, if you do projected density of states, these questions will be answered naturally, just looking on this image.
Yes, yes.
Is it
So
clear?
I need to check. I think so. I will check once again.
Okay.
Yeah.
So more questions to Hanmant, either in the audience or from Zoom? No?
If you can't answer this right now, that's fine, but would you happen to know the size of the space that this is, the ligand is coordinating around? Sort of like in angstroms, how far across is the sort of central area where the manganese ion is?
Miss.
I think that Hanmant has a table on this at the beginning of the talk. Hanmant, will you please bring? Yes.
This one.
Right, so is that distance to each one? Yes, it is. So the total distance across is about 5 or 6 angstroms. About 5 angstroms. Okay, thank you.
Okay, let's count down. More questions to Hanmant once. More questions to two. More questions three. Please join me thanking Hanmant.
Thank you.
We have like next formal talk is by Dr. Dinesh Thapa, but before this, let's David Brockner give a very brief update. I think it will be 1 or 2 slides on what he did in Chicago. Right?
So here we have the poster I presented at ACS this a little more than a week ago now. So if we look at the major feedback I got for it, so the first thing is I need to look at the orientation of the x-axis for this figure specifically. And this is actually something I noticed, but it was pointed out to me by a few people. So we see that we have this yellow line off the side here toward the bottom. Based on our expectations and these figures that we see down here, which you can't really see at the zoom I'm at, but we would expect that yellow line to be on this side of the model, not this side. The major question I got asked was, is this model experimentally possible? Which I'm not entirely sure it is. But I did have one person recommend that I look at this paper because they apparently have synthesized a simpler version of what I'm working on. So not the complexity of mine where it's like an n equals 5 and an n equals 2. They're looking at like n equals 2 and n equals 1. But I haven't completely read this paper yet. The other thing that I was getting from a lot of people was why I was using chlorine instead of bromine or iodine. And my main response was that I am transitioning to making models with bromine and iodine. And the reason I use chlorine is very simply, that's what I was provided in the beginning.
And
Well, you can't say that.
and you can say, you can say that with chlorine, the cell is a little smaller and quicker to compute.
Yeah,
And the gaps are more like,
I did not tell them that's the reason.
okay,
That is the reason I did it. I had one person who was very adamant that non-radiative lifetime or dynamics or whatever you want to call it was very detrimental to everything that they were doing. But that's because they were focusing on photoluminescent properties. So they were wondering why 2/3 of my poster, if not more of my poster, is on non-radiatives and charge transfer, where the only radiative is this corner. And for the handful of people that asked about future directions, they were wondering what pole runs, what we would expect pole runs to do for us, which I mainly talked about how they can be IR emitters, allow for IR emission. And then I just got a lot of basic questions like what is PDOS? How does a photovoltaic cell work? I had one person that literally had me walk them through a photovoltaic cell, like how the diodes work and how current works and those were the major things that I got from comments off during my presentation.
Okay, yeah, many thanks for sharing. Anyone has comments, questions, suggestions, David? Questions ready, but thank you very much. And probably announced talk by Dr. Dinesh Thapa. Dinesh, you can share your screen. The floor is yours.
Thank you so much for attending my presentation. And the topic is N79 dye functionalized PBSE, CTSE, quantum dots, structural, electronic, and optical properties. And Steven and me were doing together for this project. And I got all the structures from Steven for this calculation. So why N790 indium dye? It's one of the most common high performance dyes that are developed for solar cells, DSSC solar cells, like sensitized solar cell. It has increased stability and increased electrostatic binding and anti-filling and increased oxidation potential, which leads to deeper homogeneous levels and enhanced stability. My case studies are gadolinium selenide, with selenide, quantum dots, intrinsic or crystalline without dye attachment, and with dye functionalization. And I have 5 models for dye functionalized gadolinium. One is one oxygen atom of the dye attached to gadolinium surface of the cutie. And C2O means 2 oxygen atom from the dye attaching to gadolinium surface of the quantum dot, and 1 oxygen atom from the dye to lead surface, and 2 oxygen absorbing or close to surface of quantum dots on the lead side, which is AB2O, and PD or CD interface where 1 oxygen atom were absorbed on each side of lead and gadolinium. This is my optimized geometry for quantum dots, pristine or intrinsic. The diameter is around 1.8 nanometer, and the gadolinium and lead atoms are 31 and 33 respectively, and selenide and atom with 31 plus 33, as you see here. And on the right side, I have the optimized geometry for gadolinium 1O, which I already explained, that 1 oxygen group attaching to the gadolinium surface, as you can see in the diagram. And the hydrogen is replaced from this bond, and it goes to the surface of the quantum dots. And the right side is the schematic representation, and the sulfur of the dye, there are 2 sulfur atoms, which is represented by this yellow color, and 1 sulfur is absorbed with the cadmium of the dye, and the another sulfur is free on the dye molecule. This color representation, I just attach the charge transfer value or oxidation state for its atoms, which are close to the absorbing site. And this color, red color CD, which is this near to the sulfur or making bond to the sulfur, which has plus 0.74, and cadmium, which is close to the cadmium, which is close to COOH group actually of the dye, and it has crossed 0.93. And similarly, I have shown in the color and the event representation with your charge transfer value in this table. And I found the blue hydrogen here is has normal charge charge transfer of +1, and the hydrogen which is separated from the bond of COOH group, it has negative -1.12. It is gaining the charge from the quantum power. In general, but in the pristine case, we found in the very left side cadmium, lead, and selenium. It has a +1.65, but cadmium generally it has +2, but quantum dot it is showing the reduced value of 0.6 means during the quantum dot reaction, it lost most of its charge. This is second case, Ct2O, in which 2 oxygen group is absorbing on the surface of the cadmium side, and the very right is the relaxed structures and the schematic representation of the dye structures. And the cadmium, here we can see after relaxation how the cadmium and the sulfide oxygen are positioned by themselves, as you can see in this schematic representation. Then also I have calculated the charge transfer in its gases which are absorbed or interact on the surface. Cadmium is +0.80, sulfur +1.26, similar to the hydrogen, here COOH has +1. And another is lead 1O, in which 1 oxygen group or COO group of the dye is absorbed on the surface of lead. And I have with color representation of this element, we can see the charge transfer value for each case. Here also we can see the hydrogen atom, which is separated from this bond as the negative charge, but the hydrogen on the COOH group is still maintaining the +1 charge. This is lead with the 2 oxygen group. As you can see the relaxed structure and geometry of this model. And I have the charge transfer value for its case. In this case, this O group has -1.91 and and sulfur +1.27, and hydrogen still it is separated from the quantum dots. It has -1.0, which it is getting charge. The last model I have with the lead and cadmium interface is with 1O group on its side of lead and cadmium, and we can see the 2 oxygen from the COOH group making a breeze over here. And this is also doing the same thing on the other side.
Dinesh, just a second.
Yes, ma'am.
I have a question about your structure. So just looking on your molecule, right? I'm completely confused.
With which number?
I mean, like the molecule with NCS, with the isocyanide group, right? So the isocyanide group doesn't have hydrogen, right?
Yeah, this does not have it. Can see your
But in all cases, your sulfur is positive.
sulfur.
I'm
Yes.
not sure, like chemically, it makes no sense. Your sulfur should not be negative for this molecule. For isocyanide molecule, of course, sulfur should be negative, not positive. It's more electronegative element comparing to carbon. Okay, but just to be sure, you don't have protons on a sulfur, right? Yeah, so I'm just a little bit concerned about the charges on the sulfur. They look suspicious. You expect to have some more negative charge on nitrogen. You expect to have positive charge on carbon and you expect to have negative charge on sulfur. So it would be interesting to check the charges with the DFT calculations. Maybe you need to talk with Steven and ask him to provide the information on the charges for his case, for the Gaussian calculations.
Yes, sulfur should become negative. In general assumption. So I need to check it again. I calculated total inertia and binding inertia for exotic models, and I found that PB10 is more stable structure than others. And it has the binding inertia of -1.3, which is more negative, so it's more stable. So means the dye molecule is more bound to the surface of this model. On the basis of structural stability, I have this order. So it10 is the most stable, and PB or CD interface with 1 is stable. And negative arrow binding inertia in all the cases, which is suggesting the dye molecule is strongly bound to the proposed UV surface.
Except your interface case, because interface case is much low value.
But still negative, it is.
Yeah, it's still negative, but again, if you compare, if you think that you have these possibilities either to be attached this way or that way or that way, right? So of course, it would be more probable to be attached with the lowest energy as possible, right? So your interface attachment is really unlikely in this case, but I think it's not because of the interface. It's probably because not you, I understand that you were taking the structures from Steven. Instead of placing the molecule so it would attach in with carboxylic groups on cadmium and lead on the interface, but with carboxylic groups, it looks like it's attaching it one carboxylic on the one side, but then isocyanide through the sulfur, right, to another side. And probably because of this, you're using isocyanide groups instead of carboxylic group as linking group to the surface. And this reducing the binding energy significantly comparing to the cases when it is just attached through the carboxylic groups. So I don't think it's interface, it's really the way how it is attached through the molecule side. And again, my suggestion would be ask Steven whether he has a structure with both carboxylic groups interface, but when it's attached to carboxylic group on cadmium side and carboxylic group on the lead side.
That should have been the structure I gave to Dinesh.
Is it the CD10 and PB1?
Yeah, the PB/CD10 should be where it is attached to one carboxylic group on each side of the interface.
I think it should be 2 or, right? You probably should give him 2 instead of 1 or.
I'll check the structures that I sent to him and make sure, but yeah.
Because, maybe it's, just a second, it's not I saw this interface with the cyanide group. It's lead 1 or through the isocyanide group.
Yes.
Can you return back to your very first slide? I mean, when you just started with the structure, because I think you started with this interface, right? This is interface. Return back. Yeah. On this slide, you're showing your interface.
One is the sulfur. This is
Also, it's not interface, it's cadmium 1 also, it's on the cadmium side.
It's on the cadmium side.
Yes, this is the molecule in this one is spun around, so it's closer to the interface, but it's still on the cadmium side.
Okay, then you're not. I think it's different from Steven. My understanding is that your structures are different then. So your notations are misleading because in this case, you clearly showing that the bonds is happening not only through the oxygen, but also through the isocyanide groups, through the sulfur, right? I mean, because I think in Steven notations, cadmium 1 or when he has only carboxylic group attachment on the cadmium side, but no isocyanide is involved. Okay, So then I'm wrong. Then what you call 1O is also both cadmium 1O and lead 1O include also interaction through the isocyanide group, right?
In CD1O it is true, but PP1O.
Yeah, both. And the interface has carboxylic group attachment on both sides.
So, yeah, one sulfur and one oxygen on the cadmium.
Yeah, can you return back to your binding energies then? Okay, then trend looks like it's opposite. When you have oxygen like cadmium 2O showing minus 0.3 and it's through the two oxygens. I'm not sure why then cadmium 2 lead 2O showing well, okay.
CD1O and PP1O has the same geometry, but CD2O and PP2O, they have not same geometry. PP2O, two oxygen making a bond with lead. But in CD2O, only one oxygen is making bond with cadmium.
It is protonated. I see. Yeah, so we can explain the difference with a cadmium. Can you return back to your binding energies?
So
Okay. Yeah, so the trend with cadmium, like the difference between cadmium 2O and lead 2O is because lead 2O is having hydrogens on the surface, while Cad 2O has hydrogens on the carboxylic groups. And we can explain the difference in the reduced binding energy when it's having protonated carboxylic groups.
yes, so in this case, in this configuration, we are getting lower.
Okay, so we can contribute this change due to the carboxylic, due to the protonation processes, and with the interface, since it's also attaching through the both carboxylic groups and it's deprotonated, right? For the interface attachment. Surprisingly, your binding energy is very, very weak. I'm just trying to say that you need to kind of make some, if you show in the data, please analyze them. What's the reason just to give us the numbers? And then you make in a conclusion that it is bind to the surface. The difference in energy, like orders of magnitude difference in your total energy, how it can be exactly the same result. So definitely the binding energy is sensitive to the way how the molecule is attaching to the surface. It's sensitive to what kind of linkage group is involved, protonated, deprotonated, is it one carboxylic group, two carboxylic group, isocyanide group, right? And it is sensitive to the being interface versus lead or versus cadmium side. I would say Cad and lead side with 1O looks not very different, but for the interface, your binding energy is very weak in this case. Yeah. Which again gives you some ideas that you probably, well, if your data are correct, that the interface is probably unlikely happen. The molecules will probably bind either lead or cadmium side through one carboxylic group, because this provides the lowest energy. And again, if molecule is protonated, if carboxylic group is protonated, it's also reduce the binding energy, which means again you probably unlikely have very stable structures with protonations when proton stays on the molecule. So likely molecule is deprotonated and then it's attaching to the surface, because thermodynamically it's provide much low energy. And again, your energy is really different. 0.05 and 1.2, right? So it's orders of magnitude difference, almost more than orders of magnitude difference a little bit. Okay, and again, would be interesting to compare with the results from Stephen. I hope he already has this data on the binding energy in a vacuum and also in a solvent, because in WASP we cannot include solvent media, right? but we can look at on the Gaussian calculations just to see whether we have the same trends or not. But be sure that data from Steven showing exact, like your geometry, you're really talking about exactly the same geometries, and your notations are exactly the same.
Yeah, how to differentiate in this case the CD2O and PB2O?
Mhm.
Because their binding atom has different configuration. CD2O only.
Yeah, and another problem I think is positive charge on a sulfur. It looks very suspicious
Yes.
because it's positive in all cases. I mean, in all cases where you show in sulfur, it is positive.
Sometimes it may happen because of low number of single potential valence electron, which was from the car file. I may have to choose the single potential larger number of electrons.
Well, in any case, we need them to look on the charge on the nitrogen and carbon, because it looks like your nitrogen now should have very negative charge, but sulfur is more electronegative than nitrogen, I think. Well, maybe not. Nitrogen is more electronegative than sulfur, but carbon for sure is the least electronegative among both. So it's really strange to have positive charge on the sulfur.
So I think I need to change the pseudo potential file for sulfur.
Well,
Yes.
let's take a look on the charges on your nitrogen and carbon first. And again, compare with the charges from Steven data.
This is the better method of charge calculation. We may have another options taking charge from the atomic spheres on the outcome. We can compare both the results for this.
Maybe, okay. But anyway, so this looks a little bit suspicious and yeah. Continue, please.
Yeah, in this slide, I have the charge density difference for the total structures, quantum dots and dies, subtracting the charge density of quantum dot and charge density of die molecules.
And again, Dinesh, when you subtract them, you keep in the same geometry as it was with the interacting case. You didn't optimize each of these isolated cases.
No, we don't need to optimize.
Okay, so you have quantum dot, the pristine quantum dot and the quantum dot with a molecule has exactly the same geometry.
Same geometry.
Okay, makes sense.
Yes, so this is the 3D isosurface plot. And I took some slices of 2D structures from the adsorbed positions of the isolated hydrogen here, and 2 oxygens and cadmium plane, and from another plane for the this sulfur here, sulfur and the cadmium, and we can see the negative charge on this isolated hydrogen. Red is the charge gain, and charge gain is a negative oxidation state. And blue is charge lost, and charge lost is a positive oxidation state. We can compare this qualitative picture with our quantitative result before slides. So this atom hydrogen is suspiciously again getting the negative oxidation state because of large charge gained with the red region as shown in this slide. And zero, this green region is 0. And again, we can see in the plane oxygen and cadmium, the charge transfer between the oxygen and the cadmium. Oxygen atom getting more charge, and the cadmium losing the charge, but the charge depletion occurs between these 2 cadmium and 2 oxygen atoms. Similarly, we can see the charge gain for the cadmium and the sulfur. It is usually significant. And for the CD2O also, this is the 3D view for the charge density difference. And I sliced the 2D plane for the cadmium and 2 oxygen atoms. and significant charge depletion occurs in the bond between cadmium and oxygen. Maybe similar trend occurs in all the models, and this is another for lead 1O. And here also we can see the same pattern for the charge lossing and charge gaining case. This is orbital charge density, and for the pristine, this is HOMO -1, and this one is HOMO. HOMO and LUMO, LUMO +1. And for the pristine or intrinsic case, we see that HOMO charge density is mainly localized on the deep selenide surface. And for the LUMO, it is also spreading over the cadmium selenide case also. More on the deep selenide surface. And for the LUMO +1, we can see the charge is localization of the lead and cadmium both. So in all the cases, we can see the dominant charge density is on the di molecule for the HOMO and LUMO. And
for this case, your pristine is also preserving same geometry, or it is optimized geometry?
This is the optimized geometry.
Okay, I got it.
Each of them are optimized in each case,
Mhm.
borrowed from the other one. And this one is for the lead 1O, lead 2O, and interface case. And again, we can see the charge density localized on the HOMO and LUMO for the lead 1O, and only on the QD surface for the LUMO case on PB2O and interface case. And this one is for the +1. Let's see the total and partial density of states. I plotted the projected density of states for the QD as a whole and the diet as a whole. And this black curve is the total density of states. And in this case, we see that the HOMO and LUMO are mainly contributed from the di molecule, except in case of PB2O and PB or CD2O interface. The LUMO is mainly contributed from the PB quantum dot. This is electronic energy levels, and we can see the quantitative value of band gap in this case. And the smallest one or more rate frequency is for the interface case, which is 0.1 getting lower. And in case of di molecule and QDs, we have the more basic for the CD2O. Here I calculated the absorption spectrum.
Dinesh, just a second. Can you return back to your this representation? So, am I right that again, if I remember correctly, you projected those? Am I right that these well-splitted orbitals in the occupied levels in the valence band, in the case of cadmium 1O and cadmium 2O, they are coming from the molecule, right? Yeah. Actually, they all should come from the molecule, but in case of 1O and 2O, they are very separated from the main band. They're good separated from the main band.
In this case, HOMO is more localized, but the LUMO is not.
Yeah, I'm talking about occupied orbitals, right? About HOMO and whatever. Okay, good.
Here we can see the absorption spectrum and oscillatory strength for the intrinsic case, and for the cadmium with 1O, cadmium with 2O, and lead with 1O and lead with 2O. Here we can see the high states at the for the CD2O at this energy level. And
What's the origin of these transitions?
sorry, say again.
Well, these redshifted transitions, let's say if we compare CAD1O, you were showing that HOMO was sitting on a molecule And Luma, I don't remember. Luma probably was also sitting on a molecule. So does it mean that this redshifted transitions just coming from the molecule?
Yes, I think so. What in CD2O and this interface, they don't come from the molecule, but in other case, they come from the molecule.
Yeah, and of course, then if it is just transition from a molecule, then it is, of course, we vary off with energy, because for this molecule, I think the transition should be in a visible range, right? 0.7 is definitely too much redshifted, so the functional definitely is not the right functional for this case. And also for the pristine quantum dot, well, for pristine quantum dot, it's hard to say because it's confinement effect, right? And since we have lead selenide is known for small band gap. But for molecule, for sure, it is very unrealistic results.
Yeah, it's conclusions for my presentation. It's for structural stability, we see more stable as D214. And the negative value of binding energy suggesting that molecule is strongly bound to the QD surface. And a significant portion of spin polarized surface at the site of adsorption. And orbital charge densities are localized mainly on die, accepting D2O CD interface with charge density on gluon is localized on CD. And the red silicon interface and cadmium is smaller than other case. This ends my presentation. So,
Okay, please join me in thanking the speaker. Yeah. So, any questions in the face-to-face audience? Any questions in the Zoom audience? So I think.
Yeah, I have one. I apologize.
Yes, go ahead.
Yep. So can you explain like the overall purpose of this again? And then I guess there was a side question for Kalina, or Dr. Kalina, because she had said it's not the right functional, and I was just curious what she would recommend as the correct functional then.
Hybrid functional would give you, of course, much more blue shifted spectra.
I have not tried here other functionalities.
But first question of me was, what is the purpose of all this? How would you answer him?
Oh, this, yeah, it has many purpose. You can use in the solar cells, photovoltaics, and as a biomedicals. Because of its ruthenium dye, it has so much.
No, I think his question was not about how you can apply the system, but I think his question was more directed to your research or to your results. So what is the purpose, or probably what is the main questions you wanted to answer with your research? Am I right?
Yeah, you probably worded it better than I did. It was just like you know, examples like, what is your hypothesis for this experiment that you're doing? And if it's still under development, that's fine. I just want to know what your general idea is.
What is success?
What did you want to learn from these calculations? What questions you wanted to answer with these calculations?
Yeah, I can. My goal is to see the electronic and optical properties basically. Molecular 719 of the QD surface. I'm still looking what I can explore besides this. So it is a question to me as well. And
So, okay, you said you want to see the electronic properties, how they depend on the attachment of the dye, right?
dynamics,
Yes.
properties. Up to now, I'm thinking in that way.
But then what is your conclusion? So how does it affected by the attachment of the dye? Do you see any trends like this attachment, that attachment? What exactly is the effect on the electronic properties? Does it really have any effect on the electronic properties?
In electronic properties, yeah, definitely have lower band gap.
Okay, you're just talking about the band gap. Why is it important?
The trapping of the excitons or something. So it's a low band gap. Exciton trap is, we can analyze the exciton trap.
But what's the origin of this exciton? What do you want to excite? The dye, the molecule, or the quantum dot? Is it important? Why is it important?
Yeah,
Or it doesn't matter where you excite?
dye is more important.
So you want to excite dye? And then for what? What is the purpose of exciting? Like you have a system where you have the dye and the quantum dot. You said my excitation goes to the dye. And then what? Okay, you excite the dye. In this case, why you don't just take dye by itself? Why you need the quantum dot? What's the reason to put them together? And how this reason is aligned with your electronic structure specifics? Well, can you return back to your projected doors? So any reasons why it is important to know where the dye and where the quantum dot states are?
Yeah, dye is for the, as you can see, the dye.
Yeah, so your lowest energy excitation in this case would be probably dye excitation, as we said, like it is too much redshifted comparing to experimental data on the dye, but it's probably because of the functional. But again, so what? You put the dye on the quantum dot, and what do you expect? You want to increase optical activity of the dye for this, or what's the purpose to put them together then?
With the help of dye, we can get a more red thickness. And the more bound excitonic effect.
Well, excitons, if it is molecule, there are excitons, they're already bound. There is no such thing like more bound exciton in the molecule, they are bound, that's it. So there are very strong excitonic effects in the molecule. So again, I still don't understand what's the purpose to put these two systems together.
Let me suggest a little game or exercise so that not the author of the talk, but others will try to answer this question. So please think about your answers, raise hands if you want to. And Mead, since you asked this question, maybe you have a hypothetical answer. How do you think what the system may be helpful for and which properties are important?
Well, from the presenter's perspective, like just I can tell maybe this is new or he's just trying to understand the basics of what these types of experience are doing. From his perspective, I can see just getting the basics down of, okay, if we do this, let's say change in the system, we'll see this change in electronic information, for example, density of states, location of these. But regarding its actual purpose, I'm still in a little dismay myself. So I don't have a specific answer regarding I don't have an answer that would satisfy, let's say, Kalina's or Dr. Killen's purpose or hypotheses that they're looking for.
But you already contributed significantly to this discussion. You asked exactly the right question and very important question. If this was already explained by the presenter, right? There are no need in these questions then. But it was definitely missing in the presentation, and it's a very important question. So what exactly you want to learn from this? What's the purpose? Or what is good, what is bad? Okay, you see there is some difference when you attach in the die, the levels are kind of shifting, moving, and so on. So, but then, so what? Which of these electronic alignments is beneficial? And if it is beneficial to why and to what? For what kind of applications maybe?
I hypothesize that at least 4 individuals may give an answer. So Adam may try, Amir may try, London may try, and David may try. So any of you wants to be a volunteer?
And Stephen, I guess he's also here, right? No? Do we have Stephen?
Well, okay, so if your application is photovoltaics, then your ultimate goal is to separate charges so that you get electricity out. So, I mean, at that point, you care about where your electrons are localized because you're trying to basically, send them out different directions from your system.
Yeah, from this point of view, Landon, what would you excite, the die or the quantum dot for this purpose?
Well, I mostly did perovskite, so I would think the quantum dot.
Well, yeah. In this case, you probably expect that you excite the quantum dot, and then you expect that there should be one of the carriers, either the hole or the electron, will be trapped on the die, and then another carrier can be used to be providing the current. Well, typically, probably think about electron, right? In the conduction band can be used for running to the contacts and provide the current, and then the hole should be trapped on the die.
Yes.
And from this perspective, what do you think? Which of this alignment, if we believe that this is true or reasonable, whatever structures, which of these alignments you think will provide the best scenario for this type of applications?
Can you go back to where you're finding your I can't see the lines super well on this.
Slideshow probably will help. No? Are we good? Should we go to slideshow?
Ooh, well. Both the HOMO and the LUMO are localized on the die in this situation. What about the next set of pictures? Because that's not good for photovoltaics because then you might get recombination. Aha. So, well, okay. I guess it's backwards from how I would think you would want to do it, but the PB10 looks, you at least get some. Oh wait, no, never mind. Hold on, I'm thinking about this backwards.
Well, I think projected dose is better to look on it because then we see really the whole story, right? Can you return back to the projected dose? Okay, so let's take a look on the LED2O. So if we excite quantum dot, it means the excitation go from red to red, right? From deep valence band to, I mean, in a second peak in the, or whatever number of the peak, in the valence band around -1 to maybe around luma. This is where you excite, right? Electron is still on a quantum dot. And then the hole can move to the low states, which are located on the molecule. And if they have a strong coupling, non-abiotic coupling between these states originated from the die and the states originated from the quantum dot. So then we will have a very quick transition from the quantum dot to the die. And then the hole will be trapped on the die in this case. And electron stay on the quantum dot. So we have a charge separation.
I can't really see the plots well enough to confirm or deny, but that sounds right.
What you cannot see?
It's just bright in here, so like the yellow lines, like I basically just can't see them.
Oh, it's red and green, by the way.
Oh, sure, yeah. I'm also colorblind, so it doesn't help either.
Then you need
Oh.
to
Yeah.
Yeah, so it looks like red and green is not a good colors. And by the way, yeah, so Dinesh, don't use green and red because many people can be colorblind and they cannot differentiate between green and red.
Well, I can tell the difference between the brighter ones and the darker ones, but I can't really tell the difference between the darker ones. And to me, it looks like the brighter one is just hanging out near the bottom the whole time.
So we have a little,
Detail.
that's one. Yeah, so this is 2O.
Yeah.
So this top line that we have here, that's black, that's the total density of states.
Yeah.
And then getting real close to it, we have the first inside PTS from the quantum dot, and then we have the die. So this is an approximation, and then for it's gonna use Greek. You guys be able to see it, right?
I could probably see that. A projector in the sunny room.
Yeah, this would be what you, yeah. And this one would probably be okay too, but that one looks better.
What's the
Binding energy. It's basically, it's hanging out on here and you excite it, and then the electron gets into here, so then your hole will relax and hang out over here, and you can pull it out that end. And then your electron will be somewhere else in space, and you can pull it out that way.
Me, thank you for stimulating the discussion. Are you satisfied with the answers?
Yeah, yeah, yeah.
These are important. That's the way.
Well, again, if you want to do the charge separation, right, this is exactly the purpose why we introduce additional species to the quantum dot. So we need some kind of a thing which will be helping to separate or to break electron hole pair, right? So that one carrier will be staying where we excited and another carrier goes to another species. It can be useful for photovoltaics, can be useful for catalysis, for example, and so on, so on.
So criterion of success for this part of the work is to identify configurations that would provide charge transfer excitation. Would it be correct? Okay, let's check if there are more questions from Zoom audience. If no, let's quickly glance over typed questions and approach the glorious completion of the mission. Huh? Yes, okay, yes, thank you. And David was,
You skipped mine.
you skipped.
Oh, we would never forget about it.
Schematic structures will change. Corrections. One book cases. Yeah, after relaxation, what I got.
Dinesh, could you please return back to your slides?
So the picture's a bit small for me to actually be able to see it. But it looks like where you have the NCSs, they should be flipped. So the cadmium should be on the bottom and the non-cadmium should be on the top in your schematic diagram.
Right, you have them going to opposite ones.
And you do this for both the cadmium 10 and lead 10.
Okay.
That-
Both case.
It look, at least that's how it looks to me. Someone, you should check with a better view of the model because I had to stare at it quite a bit to try and figure that out. Because it's something just looked wrong to me, so I stared at it a bit.
Yeah, I also was confused with the structure because I thought that this is an interface. So that for better, rotate it a little bit more so we really can see that this is cadmium side. For lead is okay, yeah, for lead no problems. For lead we see that it's attached to the lead. But for cadmium side it was a little bit complicated structure, so it's unclear. Is it the H or what is it? Yeah, looks like it's almost interface. Actually, another question to you, maybe not to you, wherever you took, like if lead, for lead case, it's really sitting on a deep lead side, right? This what you call cadmium, it's really placed more like to the edge to me rather than completely on cadmium. Is there any reason why this was so different between these two cases?
Share the question again, which, yes.
Okay, I guess no reason, it just it happens, right?
I go to our question.
I don't mind.
PV2 and PV or CD2 flow provide lowest excitation from time. Other configurations provide time lowest excitation. What is the reason? How do we assimilate that different binding pattern changes the character of the lowest excitation? I need to source this answer.
I don't know the answer, but probably this binding pattern somehow affects, shifts total all energies of die and make relative changes of their energy values. But it would be key part of your work if you find this answer.
Yes.
So more questions to Dinesh? More questions one, more questions two, more questions three. Please join me thanking Dinesh again. Thank you very much for all your inspiring talks and discussion and all contributions. So next time when we meet, it will be much briefer. By now we have only one activity on the agenda for September 6th. So we will provide the abstract for the conference and we will do collective review. If there are volunteers to speak, you're welcome to do. If there are no volunteers, you just leave much quicker than today. In 2 weeks, it would be nice if Joseph will make his first attempt to talk about excited state dynamics, and there will be chances for one and more to talk if you want. If you don't, and then no plans for September 20th. You'll find volunteers, and September 27th we will need to do practice talks of posters and presentations for everyone who goes to Rochester. Plan on this. So and it's not my part, but I believe all attendees of this meeting are also welcome to join the meetings of Professor Kilina on Fridays. If you like this activity of listening and asking questions. And meeting is done. Everyone is welcome to disconnect and depart. If there are any aspects to discuss one-on-one, feel free to stay. I'll be here both on Zoom and in the room. But meeting is done. Thank you very much. It was very inspiring.
I have a question, Dr. Killen.
Yes, please.
It was regarding the ACS presentation in the future. Is that a possibility?
I saw your email, so I cannot tell you the answer without consulting with your advisor.
Of
If
course.
it's to send you to this conference, I will take 50% cost of it, but I cannot approve or not approve your travel without your advisor.
Yes, of course. I just wanted to hear what your thoughts. So no worries. And I understand it's a long time away, so no worries. Okay.
I just don't want to steal any of his rights and privileges. So
Of course.
if he decides, I will support. If he doesn't decide, okay.
Yep, no worries. I'll just talk to him and no problem. So yeah, I just had your meeting first this week. That's all. So.
Okay. David wants to ask you something.
We're
David.
meeting on Thursday, right?
Yes, I have. I have it. Multiple alarms. Yes.
Okay. I'm just screen sharing for the jobs on NERSC. So there are 5 premium working on, 1 regular going on, 2 regular are scheduled to be started in a couple of hours, and 2 dozens of regular jobs that are waiting for their chance to start. I know that for some of you it is annoying and challenges your human patience.
Dr. Kilin, I have some questions regarding the MD simulation.
Regarding what?
Regarding the MD simulation. So, as you suggested that I have to restrict because the molecule is expanding. So you said that I need to restrict the boundaries for the molecule, so it will not move. So why it is like that? Means why do you don't want to molecule should be expand?
Okay, thank you for the question. Let me speak. As we discussed with your advisor, the plan is to have several papers, right? For the first one, the goal is to just show difference of this octahedral and tetrahedral or whatever it is, and maybe add different functionals. So no dynamics at all. And you almost have enough data for the first paper. Maybe you may add just take optimized geometry for n up down equals 3 and explore it with different functionals, and then it will be more than enough for the first paper. So don't worry, this dynamics thing shouldn't delay you from working towards publication. Second, your actual question. In your realistic model, in the real life, the reason these biological tissues are not evaporated, all right? They are solid, liquid, and they have very much known density, approximately equal to the density of liquid water. If your model is expanding, it means it becomes fluffy, it has some empty spots, and it means its density goes to approximately 1 to approximately 0.3. So it's not right. It is what would happen to an astronaut who is left without a space suit in a space, just exploding body, right? Yeah. If you don't want it, it will be a little misleading. The result will be not trusted. So if there is a chance to correct this error, it would be better to keep the model as dense as possible. And one can either by having no vacuum, because vacuum is exactly you are throwing your biological tissues in a space, or in a vacuum chamber and it expands like a popcorn. Yeah. Which is not good. Or fill anything above, below with high density water molecules, and then again have no vacuum. And then this water molecule will compress it and prevent it from expanding. But just no vacuum probably is the best and quickest solution. And then after you redo with this compressed model, you redo everything like ground state, excited state, molecular dynamics, and then couples, which slightly delays your progress. But as you are getting more experienced, the final time when you get results will be not much delayed. Because for the second time, you do things much quicker.
Okay.
That
Okay.
makes sense.
Yes, and so do I need to when I preparing the model that time, instead of what you can say giving this more space, I just miss more dimensions for the vacuum or for the model.
Let's say when you do think to wasp,
Yeah.
you told that you're using think to wasp 666.
Yes.
The new model you just do think to wasp 222 or 111. That's-
Okay. Okay. So it may still the thing is what if you check the what you can say model, it is not that much dense. Maybe because of it, even if we restrict the what you can say, even if we prepare the model for 111 or 222, still I think when we will do the heating, I think it will it will not expand that much, but still the what you can say the residues or atoms will move inside the what you can say inside that particular the model which the what you can say in that particular cube which we created for the model.
Mhm.
I think so.
So
So should I add water molecule or something or?
if you can do it in parallel, because we want to have this project completed before you have your doctoral defense, right?
Yes.
There are many ideas to implement, but they may delay. So if you can quickly add water molecules and run things in parallel, go ahead. But it will be more expensive. So your system is already rather big and takes time. So
Yes.
do not select either one. Do both. Do not cancel just vacuum model. Do it at first because it will be quicker.
Okay.
And if we would try to publish what you have right now, any reviewer would ask why the model is expanding and would make some acidic, skeptical comments.
Yes. Yeah. Okay. And the question is regarding GGA U. What is the U value? I just checked. It is equal to 2. So, so I think it is a default value that one that program have, and I didn't check that one when I was running. So sorry about it. And another question. So when I checked the coupling simulation, so I made one mistake in the in-car file where I selected the NPAR value is equal to 24. I just changed that value is equal to 60. And for that value, I think there is one simulation which is actually running on the premium queue. And for it, it is creating the coupling files. And also I checked the size of it. It is more than 33 byte, means it is near about 6,000 something else. So maybe I will send you the directory for it. Can you please check for that one also?
Okay.
So maybe I will get the idea that only because of that particular error, I am getting the what you can say. The program is not functioning. I think so. Then.
Okay, I will have.
Yeah.
NPAR tag?
Yes.
Okay.
NPAR.
Yes, NPAR. So that I think when I check the error, I think initially something it is not something like the value which is it is not dividing the 64, something like that. That is the message. What is it getting in the slum out?
Safer to for all computational things to use powers of 2, like 2, 4, 8, 16, 32.
And NPAR on NERSC is most efficient when it's equal to the square root of the number of cores.
Okay.
So if you have 64 cores, you want NPAR to be equal to 8, 256 would be 16, so on.
Okay, and if we select the number of codes 4 or something like that, then we will multiply 4 into 64 and then we will square root of it, right?
Square root of number of cores.
Yes. Means if we are using 4 nodes and 64 CPUs, so 4 into 64 cores, and whatever value we are getting means it is equal to so square root of that value, that one is equal to n par value, right? Okay. Okay, I think I made that mistake. That's why. Yeah. Thank
All
you.
it should be affecting is efficiency,
Okay.
but it has a marked increase in efficiency when you do it correctly. Like some of my models I can't even complete if I don't specify that. Like they won't even get through one round in 48 hours.
Okay. Okay. So it's not only just.
It'll go through 10 or 20.
Okay. Okay. Thank you.
Yeah, thank you, Hanmant.
Thank you. Thank you. Thank you, Dr. Kelly.
And very nice talk. I'm gonna look forward for continuation.
Thank you.
Alan, thank you for joining. How are you doing? Alan is not answering. Yeah, how things are going? Do we need to talk about anything?
Yes,
Okay, let's start.
okay. Do you want to record them?
Huh?
You want to record this one also?
You want to stop recording? Or give you some secrets?
Not secret, but